Welcome everybody to the Harvard University Statistics 115-215, Bio 282, Biostatistics 282 class, Introduction to Computational Biology and Bioinformatics. I would like to congratulate everybody for your decision to take this class. In my study years, I just remember from both college and graduate school, even though I took many different classes, there were usually just a few that was really memorable and very useful for my later studies. And I certainly hope this course would be one of those, because bioinformatics is one of the most useful skills, whether you are working in statistics, computer science, and especially if you are a biologist. This is going to be a very, very useful skill. So I would like to start the course with a brief overview of the history of bioinformatics. There are several waves of bioinformatics over the history. And the very first wave is the protein sequence and protein structure wave. In 1955, Fred Sanger developed the technology to sequence protein sequence. So he first sequenced the bovine insulin, which later enabled people to actually synthesize insulin as a drug. After the technique was invented, many groups used his technique to sequence proteins. And every sequence they obtained, they want to see whether other people have seen the same sequence or not. Therefore, they compare their new sequence with a previous protein sequence to see how similar it is. And so there are algorithms or computational algorithms that are needed in order to compare two sequences and see how similar they are. So this is called pairwise sequence alignment. So you look at two sequences, and if the two amino acids are the same, they're great, they're aligned well. And sometimes even if the amino acid sequence is different, you could see that they have similar biochemical properties, therefore could be aligned together. And so computational algorithms such as Needle-1-Wunsch was developed in order to align the two sequences and provide a score to measure their similarity. And when I was a graduate student over 20 years ago, the first bioinformatics class started with learning Needle-1-Wunsch algorithm. And so as people were sequencing more and more protein sequences, comparing one sequence with another using this type of pairwise sequence became kind of difficult. By the way, the reason people care about aligning the sequence, first of all, they want to know whether they have seen this before, but also because if two proteins have the same sequence, they might have the same structure, and therefore also they have similar functions. And so in 1973, a project was started for protein data bank. So at that time, some of the protein structures were solved. And over the years, all the solved protein structures were stored in this protein data bank. And if you go there, you can see not just protein sequence, but there are over 173,000 biological macromolecular structures available. And so they have the kind of the 3D structure of the protein. If we have two sequences that look similar, they might have similar structure, therefore they might have similar functions. And as we mentioned, if there are more and more sequences becoming available, doing the pairwise sequence alignment will become very expensive. Therefore, there are databases available to store all the publicly available sequence or newly sequenced protein sequences in a database. And there are algorithms for doing faster search. If you have a query sequence of your new protein sequence, you can enter into this database and run an algorithm called BLAST, which will tell you all the hits, previous sequences that might be similar to your query of interest. And so based on the sequence similarity, and if you look at what is known about these other proteins, you might be able to guess the function or the structure of your new sequence. And so a computational algorithm for doing fast search was developed. So instead of aligning sequence pairwise, it can very quickly search promising seed sequences and figure out which one is worth doing a pairwise sequence alignment. And so that's kind of a fast search algorithm. Also, around 1994, people started predicting protein structures. As we mentioned before, if two sequences have similar, if two proteins have similar sequences, they might have similar structure and also similar function. And it seems that once the protein sequence is known, usually the structure is set. And so over the years, people were using protein crystallography to solve the protein structure, but this is a very laborious process. And so there are computational approaches trying to predict the protein structure just purely based on sequence. Around early 1990s, many groups started working on this problem, and they might publish an algorithm to say, oh, my new method is the best. And they show some examples where they can do a really good job in predicting protein structure. But then, all these algorithms were claiming themselves to be the best. And so in 1994, this competition called critical assessment of structure prediction was established. Basically, this is a competition that runs every 2 years. In the first year, experts would contact protein crystallographer in the world to say, which protein have you just obtained the crystal? Because it could take years for people to obtain a crystal. Once they have a very pure crystal, usually in a year's time, from experience, they would know that the structure would be solved from crystallography. And so they would get a collection of proteins, usually in the 100-ish number. And then they would ask all the computational biologists in the world who are interested in this problem to make protein structure predictions. After the submissions are done, they would close the competition and then wait until a specific time. They contact all the protein crystallographers again to say, which of these do you have a structure already? They would use the experimentally solved protein structure as the gold standard to evaluate whether the computational predicted structure is correct. And so in this process, they can see which ones are the real winners, because instead of predicting one at a time, you would usually predict, maybe 50 or 100 at a time. And so this is a really unbiased approach. And also it's kind of blind ahead of time. Nobody knows the gold standard until they have submitted the prediction. Then they can look at the results. So over the years, actually a lot of computational biologists who thought, oh, protein structure prediction was a really easy problem. They came in and, after a few rounds of this CASP competition, they dropped out of the whole field completely. And so, but then there are people who learn from their mistakes. They continue to improve their predictions, and there were kind of winners over the years. We'll show you some of the recent results later. One thing people were trying to use in order to predict the protein structure is to see whether in the protein there are different functional domains. For example, these are families of proteins. You can see here, they contain small stretches of sequence that look very similar. Usually they have similar sequence and therefore small functional domains and in similar structure and similar function. It's as if you say any kitchen would have a counter, would have a refrigerator, would have a stove and a sink and a microwave, right? So, if this protein is gonna have a function like this, they would need these different domains in place. And so a lot of times when people are trying to predict protein structure, they first try to locate these domains, and based on this, they can predict the structures better. So around the same time of CASP competition, a blocks database was established, which identify these protein domains, which are very important for predicting protein function. And so continuing on the protein structure prediction, we can see this is CASP9 in 2012. So usually after a competition, the experts will evaluate all the predictions and they will write a summary paper to be published. And so you can see here that year, the columns here includes all the structures that were solved, and they can see which algorithm predicted well or badly. And so each dot here is one prediction, and the target or the proteins that are easy to predict is on the left, whereas those that are really hard to predict are on the right. And so each target, you can see there are multiple predictions. And here shown in two different colors are the kind of the overall best two predictors. One is HH-Pred, the other is Rosetta. And you can see in the easy target, they can predict. And so usually the measurement of whether something is predicted well or not is called global distance test, which kind of evaluate, the agreements between the prediction and the truth or the native proteins that are solved from experimental approaches. And so you can see when the global distance test give you a score above 90%, the correct structure, which is drawn in red in here, versus the predicted structure, have highly similar, you can see the structure are aligned very, very well. So this is easy to predict, and at least the prediction and the correct structure are very, very similar. Whereas as you can see towards this end, when you have a structure that is really, really hard to predict here, the global distance tests only have a score of 10 or 20. And you can see here the correct structure and the predicted structure still look quite different. So this is the kind of state of the art in 2012. In 2018, a new player came into the field. This is from DeepMind. It's a Google company using AI to make predictions. They just use the existing PDB database to say, if I have the sequence and I have the structure, can I learn the rules? And so a lot of the other computational predictors at the time, they have to use years and years of biochemistry and protein structure knowledge to create the rules, whereas AlphaFold is kind of trying to learn the rules from just all the existing sequence and structure. And they joined the competition in 2018 and was a very clear winner in this competition and beat the second player by a lot. And you can see at that time, among all the predictions that it made, the average global distance test of all the structure it predicted is about 60, which was already much, much better than the second player. But what is even more amazing is by 2020, 2 years later, in AlphaFold 2, this is CASP14, the average prediction can reach about 90. Actually, a score above 90 is considered roughly equivalent to the experimentally determined structure, which a lot of people in the field consider that protein structure prediction problem on a single protein is a solved problem with AI, which is really fantastic. Of course, there are still new problems, such as when you have a big molecule which includes multiple protein in a complex, how do you really predict this big complex, and how do you predict protein-protein interactions? So those are still unsolved problems, but we are hoping, really with computational biology or AI, that over time, you can get better and better at this.